The ultimate objective of modern oncology is straightforward in theory: strike the diseased cells without inflicting collateral damage on the healthy tissue surrounding them. Traditional chemotherapy fails this test. It floods the patient's system with highly toxic compounds, destroying rapidly dividing cancer cells while simultaneously damaging the healthy structures in its path. To narrow the blast radius, medicine developed monoclonal antibodies. These are large, lab-grown proteins engineered with a high binding affinity for specific molecular markers found exclusively on the surface of a cancer cell. But antibodies carry a mechanical flaw. They are massive. Because they are highly complex biological proteins, they are physically blocked from penetrating deep into the dense, high-pressure environment of a solid tumor. They get stuck on the outer periphery, while smaller molecules slip right past. To effectively infiltrate a solid tumor, oncology requires a physical paradox. We need a weapon that possesses the exact targeting capabilities of an antibody, but packaged into the microscopic dimensions of a synthetic chemical. That paradox exists. It's called an aptamer. An aptamer is simply a short, synthetic, single strand of DNA or RNA. We are taught to think of nucleic acids exclusively as information carriers, the biological blueprints that our cells read to build proteins and pass on traits. But chemists are ignoring the genetic code entirely. Instead of reading the strand, they are using the physical molecule itself as an architectural building block. Without a matching partner strand to pair with, a single sequence of DNA becomes unstable and collapses into a tight, solid knot. By treating DNA as a raw, moldable nanomaterial rather than a biological manual, scientists can engineer physical structures small enough to slip deep into a tumor, yet complex enough to recognize a specific disease marker. The transformation from a flat line of code to a three-dimensional tool is driven by chemistry. Because the single strand needs stability, it contorts and folds back onto itself. As the sequence contorts, individual nucleotides within the strand link up. Hydrogen bonds and van der Waals forces snap into place, acting like internal scaffolding to hold the newly formed sections together. At the same time, the phosphate backbone of the DNA carries a strong negative charge. It naturally seeks out positively charged pockets within the fold, drawing the strand even tighter and locking the structure into a rigid, permanent form. Depending on the exact sequence of letters, this folding creates highly complex, stable structures like hairpins, loops, or dense blocks called G-quadruplexes. This generates a very specific external topography, complete with distinct geometric ridges and grooves. This external topography acts as a precise geometric interface. The grooves and ridges are shaped to interlock through shape complementarity with corresponding protein receptors on the surface of a target cancer cell. The physical shape of the folded strand delivers the exact targeting capability of a large antibody, but it occupies a fraction of the physical volume. There is a massive engineering hurdle here. How do we design the exact sequence of nucleotides required to fold into the perfect 3D shape for a specific, newly discovered cancer receptor? We don't. Designing that molecular geometry from scratch is practically impossible. Instead, chemists synthesize trillions of completely random DNA strands in a test tube and force them to compete against each other. This diagram outlines how that competition works. It's a process called SELEX, or systematic evolution of ligands by exponential enrichment. It starts here on the top left, where that massive library of random single-stranded DNA is exposed to the disease target. The vast majority of those random strands won't fit the target. They are washed away and discarded in the partitioning phase. As you follow the cycle downward to the elution step, chemists isolate only the extremely rare strands that successfully bound to the target protein. Those surviving strands are then recovered and multiplied through PCR amplification. This circular loop repeats over and over. With each round, the target environment becomes harsher. The chemistry mutates and enriches only the absolute strongest binders, until a single, perfectly fitted aptamer sequence emerges. This test tube evolution bypasses the need to farm antibodies in living cells. It replaces slow, expensive biological harvesting with rapid, scalable chemical synthesis. So once we have isolated this specific structural match, how do we actually deploy it inside a patient? We use aptamer drug conjugates, or APDCs. Chemists take the targeted aptamer and covalently bond a highly toxic chemotherapy payload directly to its structure. When injected into the bloodstream, this conjugate navigates through the body. It ignores healthy tissue because the healthy cells lack the specific surface receptor that physically matches the aptamer shape. But when it encounters a tumor, the aptamer targets the cancer cell's unique receptor through shape complementary binding. It docks seamlessly into place. This physical connection triggers the cell to absorb it. The cancer cell literally engulfs the aptamer and toxic payload. By utilizing this mechanism, a devastating broad-spectrum chemotherapy drug is concentrated exclusively inside the tumor, leaving the surrounding healthy tissue intact. If this mechanism is so effective, there is a reason aptamer development was delayed for decades. In their natural state, they are incredibly fragile. Because they are so small, about 15 times smaller than an antibody, they pass right through the body's renal filtration barriers. The kidneys flush them out of the blood in minutes, long before they can accumulate inside a tumor. On top of that, the bloodstream is patrolling with nuclease enzymes. These enzymes are designed to hunt down and shred free-floating DNA, meaning an unprotected aptamer is chewed to pieces almost immediately. Modern chemistry has engineered a rescue. Through a process called pegylation, chemists attach a large inert polymer called polyethylene glycol to the aptamer. This artificially inflates its molecular weight, making the entire complex too bulky for the kidneys to filter out. Furthermore, by making slight chemical modifications to the sugar backbone of the nucleotide sequence, like substituting in specific fluorine atoms, chemists render the aptamer completely invisible to destructive nuclease enzymes. Aptamers fill a critical gap in oncology by infiltrating the dense, hostile environments that biological proteins physically cannot enter. By treating DNA as a synthetic architecture rather than a biological code, medicine gains a class of programmable, microscopic treatments that are cheaper to scale and precise enough to redefine targeted therapeutics.